It's a pleasure to be here this morning and to talk to you about analyzing and interpreting DNA methylation data. I would like to start by thanking the organizers for putting together this great meeting and for giving me the opportunity to present some of our recent work. You may wonder what this Center for Molecular Medicine is about, because this is relatively recent for me. I moved there to Vienna about a year ago to start a lab that applies high-throughput technology to figuring out the functional role of epigenetics in cancer. But much of the work I'm presenting today is based on a very productive collaboration with the Max Planck Institute for Informatics, where I used to work for quite a few years together with, or in the department of Thomas Lengauer. And I'll also acknowledge the Ph.D. students who may basically continue to make this collaboration a very productive one along the way. Now, without further ado, I would like to introduce the 4 topics that I will be talking about. I will start with a very brief introduction, motivating why we are interested in DNA methylation. But I guess with this audience, I wouldn't have to convince you that DNA methylation is interesting, otherwise you wouldn't be here. Then I will talk, although this is an Infinium meeting, I think it is important to see the Infinium in the broader context of available assays and see where it fits and where other assays can also complement its utility. Then the main part of the talk will be about the bioinformatic analysis and interpretation of the data, relatively methodological approach here. And then finally, in the last couple of slides, I want to show a few applications of these kind of assays. So of course the genome, this is not just DNA methylation. There's on the one-dimensional, it's the protein coding genes, DNA binding motifs, and everything that's encoded in the DNA. There's also the epigenome. It's not exactly the second dimension, but like 1.5 dimension, with DNA methylation, histone modifications, and various marks that could be heritable over mitotic cell division. But of course, there's also the 3D nuclear organization, where different parts of the chromosomes come together to form transcription factories or to be associated with a laminar and being silenced. And of course, we always, although we look at DNA methylation data a lot, we have to keep in mind that this just shows us some part of the picture, but I would argue a very important one, because DNA methylation, in contrast to the genomic DNA sequence, is cell type specific. It is gene regulating. It has this very nice aspect to it that of mitotic heritability, where the mechanism is actually understood. The DNA being, during cell division, being separated into 2 strands, then the new strand de novo synthesized without DNA methylation, and then the maintenance DNA methyltransferases copying the DNA methylation from one strand to the other. So it's that for DNA methylation is probably the only epigenetic mark for which it is actually plausible to assume that DNA methylation patterns could be propagated in dividing cells for many cell divisions for many years and maintained. DNA methylation is deregulated in cancer, and one thing where I believe like DNA methylation has a major strength also compared to RNA based biomarkers is that at the moment in clinical diagnostics, a relatively large investment is being made in putting together very high throughput DNA-based cancer gene diagnostic services, basically. And with DNA methylation, because it's a DNA-based thing, it's relatively straightforward to basically hitchhike on this infrastructure and bring additional tests to the clinic that would be more difficult with RNA-based methods. So a few words on the study design. I think the most of the studies that are using the Infinium 450K assay are conceptually relatively straightforward. You have a couple of cases, a couple of controls, you do epigenome profiling on them, and then you analyze and interpret the data. But this relative simplicity of the study design shouldn't make people think that it's actually like a straightforward thing to do, because there are quite a few relatively complex, sophisticated points that need to be addressed to make something like this not only happen, but actually be successful and meaningful. Probably the most important is to have an actual concrete question or a hypothesis, or at least a clinical application like a biomarker, a medical question where you could use it in the end. Avoiding a situation where you basically do DNA methylation assays because you've done genotyping, and then that's kind of the next obvious thing to do. But have a more specific idea of why exactly you're doing it and what the goal is, because this influences then also what kind of tissue you would like to work on. For example, if you're looking at Alzheimer's disease, there's probably very little point at looking at a kind of peripheral blood, unless you believe that DNA methylation has already been kind of disturbed in the very early embryo and then propagated in blood and in the brain tissue for many decades. But this is something that it's very important to kind of make clear at the beginning already. What's your motivation for looking at the tissue that you have in hand, beyond that you just happen to have these samples in the freezer? Controls is also a more critical point than in classical genome-wide association studies, because there is the chance of actually using matched control from the same patient, which, where it makes sense, can substantially increase the statistical power. For epigenome profiling during the discovery phase, obviously you use genome scale technologies, but then there's also quite some value to locus-specific technologies when it comes to validating in thousands and thousands of samples. And of course, like in the GWAS field, in theory, no epigenome-wide association study should be published without replication in a different cohort for the same phenotype. That obviously being not always realistic, but certainly this should be the standard. And adhering to the standard in the standards has substantially contributed in the GWAS field to overcoming this reputation of the initial GWAS studies of being not reproducible, not well conducted, whereas large-scale replication can really boost the credibility of a field. Then in the analysis phase here, it becomes a bit very project-specific. Typically, people want to correlate their observations with gene expression or cellular phenotypes and arrive at some form of biological interpretation or medical utility. Second part, let's look at which assays are available and what are their respective strengths and weaknesses. So Peter Laird very nicely summarized the landscape of DNA methylation assays a couple of years ago in a review paper, plotting on the one hand the genome-wide coverage with low coverage on the left and high coverage on the right, and on the other hand kind of the cost structure and practicalities, how many samples you can realistically analyze. So things have moved a bit towards kind of the upper right corner here, but not very much over the last couple of years. So by and large, this representation is still accurate, with whole genome bisulfite sequencing giving you the most comprehensive coverage, but only allowing like a couple of dozen samples to be processed, and the Infineum array being somewhere more towards the center, allowing very large sample sizes, but with this coverage, like whereas here you are around 30 million CPGs, here with the Infineum assay you are like 500,000, so less than 1 by 50 of what whole genome sequencing can give you. So looking at the right side of this diagram, we are looking at assays that can be used effectively for genome scale discovery. And here, a couple of years ago, we actually decided beyond like drawing these diagrams, we wanted to actually see how these different assays perform in practice when you apply them to a defined set of reference samples. So we selected 4 assays that we felt were the most interesting at that point in time, which was MEDEP-seq, antibody-based enrichment of methylated regions, methyl binding domain-seq or methyl cap-seq, where you basically enrich with a DNA binding domain protein instead of an antibody. The Infinium 27K assay at that point, basically taking the concept of array-based genotyping to DNA methylation data, and then bisulfite sequencing. And in that study, we used targeted approach using reduced representation bisulfite sequencing. But our colleagues from the West Coast also did whole genome bisulfite sequencing such that we could also compare to that assay. This is all published, so I don't really want to go into much detail, but what you can already see here comparing MEDIP, MethylCap, RBS, and Infinium 27K data, the data look very differently. And this is also has been like a, so this is why you need quite different bioinformatic approaches for analyzing these different data sets, at least in the normalization phase. Later on, it can be kind of converge more, but during the normalization phase, you really need to understand the specific characteristics of your assay. Specifically here, in the case of MEDIP and MethylCap, absence of reads. So what's basically being plotted here is the read density in a given region. Absence of reads can mean either lack of DNA methylation, because no methylation enrichment could happen, or inability to pull down anything, because there's either no CpGs there, or because for some biases in your library preparation protocol, these regions are, for example, too GC rich to actually sequence them on one of the standard sequencing technologies. In contrast, for the bisulfite sequencing-based approaches and also for the Infinium assay, you can actually distinguish between areas that are not covered here in RBS and more so in the 27K assay, and regions that are actually unmethylated, being in light blue over here. So we compared this in terms of accuracy, coverage, and most interestingly, we felt it was for a given sequencing depth or assay choice, how many differentially methylated regions you actually find between typical cancer and non-cancer samples. So jumping directly to the conclusion of this study, for the two enrichment-based methods, we observed advantages like their high nominal coverage. So in theory, they are genome-wide, although practically speaking, not in all parts of the genome, you have sufficient CpG density to actually pull something down. But I guess the main advantage of these technologies is that they are relatively cheap and similar to ChIP-seq with a relatively straightforward protocol, so something that many labs doing epigenomics are already somewhat used to. The disadvantage is this process of quantitative enrichment of certain parts of the genome, which is very difficult to normalize and very susceptible to various forms of bias. So if you're working on a MEDIP study or a MBD-seq study, it's always a very good thing if a single person can do all the pull downs. And you can also sequence them within a relatively short time period, because any sequencer update in between makes your data completely incomparable, because in every sequencing update, which typically you don't have under control if you sequence via a local sequencing facility, if they switch their kits or the way they are hybridizing the flow cells, then the GC bias tends to change, which basically changes your DNA methylation results. So these are more assays that are more suitable for relatively compact studies done over a relatively short time by a single person than for large kind of consortium-style studies. Reduced representation bisulfite sequencing works very well in our hands with more limited genome-wide coverage of 2 to 6 million CpGs. This really depends on how you do the size selection, how many restriction enzymes you use. So you have some flexibility there, but you cannot say, I want to have this particular CpG or that particular CpG. So you're a bit dependent on where your restriction enzyme cuts. But at the same time, you are like 4-fold to 10-fold higher in terms of genome-wide coverage than with the Infinium array at a very similar cost range. And you have the advantages of having a sequencing technology. Then the Infinium assay, which is the focus of today's workshop, has what we see as its advantages are the good accuracy and the similarity to genotyping. That's probably the biggest strength. If at an institution a high throughput GWAS pipeline is established, it's relatively straightforward also to conduct Infinium studies on a similar scale with hundreds or even thousands of patients, which is something that at the moment would still require either collaboration with some people who have this all set up or a significant investment in terms of kind of optimizing protocols to do this kind of scale with a different technology. So, this is also why the Infinium has been used or is used. I guess the bulk of Infinium arrays that are being run are run in relatively large studies in cancer genome sequencing or in cancer genome methylation profiling, or in particular in epigenome-wide association studies where cohorts that have been analyzed using genome-wide association study microarrays are now being processed with the next generation of assays. One limitation that does have caused a bit of trouble to some of us is the restriction to human, which makes it impossible. Like for basically, if you want to do mouse, you have to use a different assay. Simple as that. And then whole genome-wide bisulfite sequencing is actually quite complementary to the Infinium assay, because you could initially profile a relatively large cohort with the Infinium assay and then select like a relatively small number of the most interesting cases for truly genome-wide deep coverage sequencing. But if at the moment, if we do whole genome bisulfite sequencing at a coverage that we feel gives us at least some level of quantitative resolution, it still costs several thousand euros to do on a single sample. So it's still a very costly assay. So in addition to these mapping or discovery cell technologies, there are these validation or biomarker technologies that you can scale to a much larger patient number, but they are more restricted to one or a handful of genomic loci. These assays are very important if you want to eventually move your assay as a biomarker into the clinic or profile like very large validation cohorts. So building, and there the set of the choice of validation assays is even more diverse, and we were wondering what are actually the best of these assays. Here comes one other thing into play, and namely the question of robustness and interlaboratory reproducibility. So we decided to deviate from our previous technology comparison and not again do everything in our lab, but instead do it in a decentralized way. So what we've done now as part of the Blueprint project, we have collected reference samples, a set of 22 mandatory and another 10 optional reference samples of different characteristics. We've done comprehensive characterization of these cells and then, or these reference samples, and then identified 16 specific loci that then a number of participants from the community are currently starting to analyze using their favorite assay. So from basically we have for each of these reference samples we have 40 tubes with 1 microgram of DNA, and these 40 sets of 40 tubes each and sets of 22 mandatory samples, 1 tube each, are basically sent to participating labs from Blueprint and beyond, who are then applying their favorite assays such as MSP, Methylite, Pyrus sequencing, Epityper, but also more recent homebrew assays like bisulfite, kind of PCR-enriched bisulfite sequencing on the MySeq and similar assays. And then we are basically all the data is being sent back to us, and we perform an integrative assessment of data quality of all of the assays in terms of accuracy, reproducibility, sensitivity, specificity, but also aspects such as cost structure and scalability, which will then be reported in one analysis paper, but also back to all participating labs so that they know how their assay works compared to everything else. So this has, as I said, this was originally started within Blueprint, but has gotten much broader attention now, such that we already have 20 labs at the moment participating, which are really some of the leaders in the field who will contribute their assays that they use most often in high-throughput validation of specific loci, and a few assays that are just coming up. So if any of you has an assay where so in theory we are kind of closed and starting, but if any one of you has an assay that you feel really fits here and probably no one else is contributing, or you think you're or your lab is the person who's most experienced with that assay, we still have a couple of tubes left that would allow us to include a couple of more groups in this technology comparison. And then coming to the main part of the talk, bioinformatic analysis, in particular of 450K arrays. Here, again, the general workflow, and over the last couple of years, we've tried to put together, this is now in very close collaboration with the Max Planck Institute, a set of bioinformatic tools and software solutions that basically help you along these different steps. Including RNA-beats for infinome analysis and also whole genome bisulfite sequencing analysis, then several tools for prioritizing regions for experimental follow-up, and biomarker optimization assay software, Big Analyzer and MathMarker. So let's go to RNA-beats first. I will come back to this slide, so if you want to keep track of the URLs, there's still a chance to do. RNA-beats is a project done by 3 Ph.D. students in Saarbrücken, Jasna Asenov, Fabian Müller, and Pavlo Lutzig, who basically all of them were working on some Infineum 450K analysis, and we decided that it would probably be a good idea to share approaches to standardized technologies, etc. And this emerged over the last year into a very robust and now also already widely used software for Infineum 450K analysis, which we have recently extended now that it supports also bisulfite sequencing and essentially any DNA methylation assay for which you can infer single base pair DNA methylation scores. So even MEDIP data you can pre-process in a way that you get single CpG methylation levels and then use RNA-beats for the downstream analysis. So the scope is, especially for Infineum, is very comprehensive. It starts from the IDAT files and ends with a report of containing all kinds of aspects of the data, providing normalization, batch effects, differential methylation regions detection, etc. Everything in a very easy to use manner. Currently implemented as an R package, but a really painless R package, and with a web service also planned for like smaller analysis. So here's very briefly the workflow. You basically start with whatever input format you have, BAT files, IDAT files, Genome Studio output, tab-separated tables. You can also download data directly from Gene Expression Omnibus. This is then being normalized, subjected to quality control, initial probe filtering, detection of patch effects, methylation profiling, visualization, differential methylation calling, and then data export to a genome browser hub and Epi Explorer export, etc. Here are a few illustrations of the diagrams. And I think the best, if you're interested in this in more detail, the best thing is to go on our website where we actually link some of these example reports. So you can basically see there what RNA-beats can actually generate for you in a pretty much fully automated manner. There's also Fabian's poster here at this meeting later today, so stop by and ask him questions. We're also still in the phase where we are, so the tool has been online for like 6-7 months now, so we and we've quite a few people, maybe even some of you, have used it quite intensively over the last 6 months, so, which has really contributed also to the quality of the tool, such that we basically, based on this feedback, we tried to do pretty much everything people asked us for, unless we felt it was completely unreasonable. So you basically, or at least some of you, really helped to make this a useful tool, and we are, at least for the next couple of months, we will still be in this phase of really being very interested in feedback, implementing on relatively short notice whatever we think makes sense in terms of comments. And I think those people who basically used it, they got the impression that they were very responsive in terms of addressing problems, etc. As I said, R&Beats is very simple to install. You just run a single command in R, and it does the rest for you. Then the next step to get started is you have to put together a sample annotation sheet, but this is not something that follows like an idiosyncratic format. It can be as plain as this, and even the headings are not mandatory to use exactly like this. You just say which samples you have, what groups they belong to, and where you can find the IDAT file or BET file or whatever you use it as input. And the tool will be smart enough by itself to see that there are a couple of tumors, a couple of normals, that you may want to compare the tumors to the normals, identify differential methylation. But there's also some kind of patient ID that you may probably also want to compare tumor 1 to normal 1 and tumor 2 to normal 2, but not necessarily tumor 2 to normal 3. So it has a relatively comprehensive parsing algorithms to figure out based on a relatively standard sample annotation sheet that you may put together in Excel. And then based on that, launch an automated analysis again with a single command where you just say, okay, this is my sample data, here you find the data, run it, and then it generates this HTML reports that you can find on our website. So briefly, 1 or 2, 3 more examples of tools that we have developed. The first one is Epi Explorer. This basically addresses the question, once you have identified your differentially methylated regions, what are you going to do with them? Which of them you actually want to validate? Which of them you might even want to start overexpressing an associated gene? So we felt like what would be useful is something like a Google web search tool for genomic regions, where you can basically, following this Google paradigm of just typing something in, seeing what comes, then changing a bit until you actually, among your top 10 hits, have the stuff you're actually looking for. Here you basically upload your set of genomic regions, and then in real time, with like 1 or 2 second response times, you can basically click your way through, show me only those differentially methylated regions that are associated with an H3K27 site, methylation site, and in ES cells are located close to a Hox cluster gene and are associated with a CPG island, but at the same time not overlapping an H3K4 trimethylation site. Something like this. You basically just click them together here, and for everything that you click on, you get immediate feedback in terms of graphical illustrations. And then comes the phase, once you've basically figured out which of the regions you want to follow up on in a larger cohort, comes the next step of basically going into biomarker optimization or large-scale validation. For this, we've 2 tools online. The first one is Big Analyzer HT, which is the kind of bigger brother of the Big Analyzer tool that we've have online since 2005 for clonal bisulfite sequencing, and which is still surprisingly widely used for exactly that purpose. But now I think more and more people are switching from clonal bisulfite sequencing because they are sick and tired of picking clones to basically locus-specific bisulfite sequencing on iron torrent or MySeq technologies. And this tool basically allows you relatively effectively and very simplistically using web-based tools to analyze the resulting data. And then the last step is to really go into a very high throughput assay, for which we developed this Methmarker which is essentially an assay design tool for assays such as COBRA, basophite pyrrole sequencing, Methylate, etc., where for a given relatively small genomic locus, it really helps you to place your assay exactly where you want it to be and check its sensitivity and specificity for use as an epigenetic biomarker. So that was basically the part on the methods. So in the last few minutes, I want to briefly discuss some applications that we are particularly interested in that basically illustrate how you can use some of this in like real-world research. So one study that I did while I was still working with Alex Meissner at the Broad Institute was basically trying to figure out what happens to DNA methylation during adult stem cell differentiation. So on the left-hand side, you see the tissue differentiation hierarchy in the blood, and on the right-hand side in the skin, with the stem cells on top, progenitor cells, and then terminally differentiated cells at the bottom. And basically what we did, our colleagues sorted these cells out for us, so Derek Rossi for the blood lineage and Elaine Fuchs' lab for the skin lineage, purifying a highly pure cell populations, for which we then did reduced representation basophite sequencing. Remember, this is mouse, Infinium is not applicable. Plus, we had only, for some of these cell types, we had only 30 nanograms of DNA, which is quite feasible with reduced representation basophite sequencing, but difficult for most other assays. And then we basically analyzed the data in terms of enrichment for certain biological motifs. And it turned out that we could actually, in the computer, reconstruct the hierarchy, the tree of the hematopoietic differentiation lineage. So what I show here is the hematopoietic stem cell on top, the progenitor cells, the terminally differentiated cells. But this is not just like an illustration. This is actually a computer-generated diagram that was given as input only the DNA methylation data for all these cell types and the gene expression data of all these cell types, and was actually able in the computer to reconstruct what people believe is very close to the actual true hierarchy between these cell types. Of course, for the hematopoietic lineage, this is kind of pointless because we already know pretty much how the hematopoietic lineage is structured, but it shows that DNA methylation very closely reflects the kind of cell of origin or lineage choice cell fate of cells, and really can add something beyond gene expression. Based on gene expression data alone, this would not have been possible for us. This also converged very nicely with a project collaboration with Manel Estella's lab from Barcelona, where we looked at cancers of unknown primary site. These are metastatic cancers where you first diagnose the metastasis, for example, in the liver, but you don't know where the original cancer is in the body. So these are very difficult to treat then, but it seems that the DNA methylation can actually help figuring out what's going on in these samples. We could show by basically creating a reference set of 1,600 tumor and normal samples for various tumors. Back then with the Golden Gate assay, we could create like an epigenome landscape of cancer that would allow us to cluster these cancers of unknown primary site together with their associated reference cancers. And then we see that some of them are clearly derived from colon, others are derived from lung and breast. And this is now being taken further to clinical validation again by the group of Manel Estella. And this is just a very brief slide. I think we can take this concept of epigenetic biomarkers a significant step further and learn from what has been done very successfully in HIV therapy, coming up with a setup where we take a relatively small number of patients for which we can do high content drug resistance assays, and at the same time genome scale genome, epigenome, transcriptome characterization, put all of this together in a genotype-phenotype database and start predicting which kind of drugs are applicable for any specific patients. With that, I would like to conclude the main part of my talk, and just one summary slide. So this diagram is probably too small to read. It's taken from the recent review paper on analyzing and interpreting printing DNA methylation data published last summer. Just summing up that the step of the actual DNA methylation profiling here on top is just the first step, and as important for doing a successful study are the data processing, data visualization, and validation and interpretation. Such that, at least on the sequencing side, we're doing lots of sequencing. We have certainly reached the point where for the typical study, the costs of actually making sense of the data are higher, sometimes substantially higher than the sequencing cost of basically sequencing a significant number of patients. And then finally, on my last slide, I would like to advertise a project called GoDMC that has been put together mostly by Caroline Ralton and a consortium of other scientists. This is basically follows on a very well-established and I think very useful practice in the field of genome-wide association studies, where people who have profiled a number of cohorts in their labs at some point joined forces with other people having similar data to perform large-scale meta-studies, which can then allow you to go into completely new phenotypes or a lot deeper into the original phenotype. So for this study, we are in a first round of basically trying whether something like this is also like pooling data and expertise from many different labs in kind of meta-analysis where everyone would contribute certain parts of the analysis and certain parts of the data. So that's in the first round we are going for those data sets for which Infineum 450K or whole genome basophil sequencing data are available, but also complete genotyping data, such that we can link the genetic, basically look at the genetics of DNA methylation. With that, I would like to conclude and in particular acknowledge the team at the Max Planck Institute, with Fabian Müller actually being here, and also Yasin and Pavlo Lutzi contributing substantially to the R&Beats package. Thank you.
Okay, are there any questions?
Hi, one of the key steps that you mentioned was validation. Can you give us a sense of what proportion of targets, either gene-rich or gene-poor, that you think is important to validate after doing any of the discovery type techniques that you discussed?
Yeah, validation is an important issue. I think one concept that comes originally from the ICGC cancer genome project is the separation, or maybe even from the ENCODE, is the separation between verification and validation, which people sometimes tend to mix up. Verification means you basically test what you've already found with one assay with a different assay on the same data. So you have one cohort, you do Infineum analysis, and then in the very same samples you actually test by basophil parasequencing a couple of your targets. This helps you to avoid biases of the assay. Validation means you take the same assay or a different assay in a new cohort, basically. At least in new samples, but preferably in a cohort that was collected elsewhere, to really kind of make a general statement. And I would claim that many of the assays, certainly the Infineum 450K assays, certainly basophil sequencing when done in a competent lab, are sufficiently standardized nowadays that verification is not strictly necessary anymore. So this classical analysis of like a couple of markers by basophil parasequencing to show your assay works, I think that's a waste of time and money. What you should rather be doing is use a different assay in a new cohort to really provide robustness to whatever has been observed.
Any? Oh, yeah, oh, there.
Well, first of all, congratulations for the R&Beats package. I've been using for like 2 months. I'm very happy with it. cell type, the number of cell counts.
That's a feature request well taken. Yes, it would be very nice to support this. So just for like the wider crowd, what is a major issue in many ongoing projects at the moment is just tissue heterogeneity. So if you just take whole blood samples, which many epidemiological cohorts have been collected over the years without separation into clean lymphocytes or even T cells or even CD4 T cells, then you have the problem that differences in all of these have their specific DNA methylation profile, as we saw in the mouse work that I showed. So if you just do bulk analysis, you get all kinds of confounding factors. And there was very nice work by Hausmann, Dr. Hausmann from the US, who could show that you can actually tease this apart by using some form of principal component analysis. So that seems to work quite nicely, but at the moment is not sufficiently standard. I would say that we could completely automatize this process. It would, for blood, we could probably do it, but there's a risk of like garbage in, garbage out. If we just provide like an automatized analysis of this, and people have, if blood has been collected in a slightly different way, that certain cell types, granulocytes for example, are lost, it will, or overrepresented, then it will cause trouble. So this is something that would be interesting to add to RNBs because many people have to do it now and reviewers really push for it. I'm not 100% sure if the technology is mature enough yet to kind of get it into this 100% automatization paradigm that RNBs follows. But certainly we will discuss this.
Other questions?
Oh, right. You already sort of answered the question with this answer, but what I was wondering really is, in your mouse work you showed that it was the medium density CPGs that had the most power to show the tissue specific type. Do you think that the current Illumina array has enough coverage of these medium density CPGs to give you a reasonable power to do this?
Yes, I think so. I think for many of the things that people have successfully looked at, or like one consistent finding is that distinguishes DNA methylation from kinds of cancer genome sequencing is that like especially when you do like pediatric tumors or leukemia, which tend to have relatively few, relatively defined alterations, like a single fusion gene plus maybe a p53 mutation driving the cancer. That is something that's very specific, and if you have the fusion gene not covered or the p53 mutation not covered, you miss out on something. For DNA methylation, we haven't really seen this at all, that there's like a single gene being methylated, whereas everything else is fine. Such that even with assays with moderate coverage, you kind of get the big picture. And I think, yes, but I sometimes people think there's something very kind of special about these regions of moderate CPG density or CPG island shores, etc. I don't think so. There's nothing magic about these regions. They just are in a range where they can fluctuate a lot. So they use the space to fluctuate. If you're sitting close to zero, like you can think of basically DNA methylation propensity. This is something we published like 2007 in a purely computational paper, that you could think of DNA methylation propensity as like a larger spectrum. You can be extremely resistant to methylation, you can be extremely susceptible, or anything in between. And if here's 0 and here's 100%, and if you're very resistant to methylation, then you just can't fluctuate a lot. Whereas if you're in the middle, there's quite a bit of space going both ways. So I think this explains to a large percentage why we see this kind of moderate CPG dense regions being the most informative.
Have we got one more question?
Just a question in relation to normalization for 50K data. RNDs, I think, uses either the Methylumi method based on Illumina's package lineage or uses SWAN. And there's been a number of other methods that have been published in the last 12 to 18 months. So I wondered if you had any comments on the relative benefits of normalization methods over each other.
Yeah, that's, so our goal for, there are basically two approaches how you can handle this. The first one is you just support all of them, which we will probably also do, but the second one is to recommend one, which helps especially people who are just starting off on this kind of thing, and who just want to have something automatically going. And so far our choice was SWAN for this kind of what we would recommend to if you just run it, but we are currently revisiting this issue to make our decision based on a couple of very nice comparisons and additional studies published recently to basically give a recommendation that we feel is robust and reliable for the next year or two. But I think the normalization algorithms at this point in time are relatively mature already, such that if we select now the best one, probably there's not much more you can gain. You can always gain 2, 3, 4% by doing something better, but I don't see like a major thing that would basically completely abolish batch effects on the infinium array. Probably something like this it would still need the batch effect removal step. So that's probably not going to happen.
Okay, if we want to thank Christoph again.